Okay, okay. So, welcome everybody to this seminar, which, as all you know, is we have Dr. Guillermo Montoya as the invited speaker, a good friend of the institute. This is the second or third time he visits us. And let me tell you just a few words about his, I would say, very successful scientific career. And Guillermo Montoya studied biochemistry and biophysics at the University of the Basque Country and got his PhD in chemistry at the University of Zaragoza. After getting this PhD degree, he went to Heidelberg, the EMBL, and also in Frankfurt at the Max Planck Institute for Biophysics. In Heidelberg, he became group leader and later director of the structural biology program. Oh, sorry. He returned to Spain and became group leader and director of the structural biology program at the CNIO. Since 2014, he is professor and research director of the Protein Structure and Function program of the Novo Nordisk Foundation Center for Protein Research at the University of Copenhagen. Well, as you probably know, his main research interest of Guillermo is the characterization, both the structural and functional, of CRISPR systems, and he is going to talk about his later result about this field. Well, his excellent contributions in this field of programmable endonucleases make him awarded with several prizes, such as the Caja Rural de Granada and the Spanish Ministry of Health, and in 2009 the National Prize in Biomedicine from the Mutua Madrileña Foundation. Guillermo is an elected member of the EMBO and received in 2019 the Novo Nordisk Foundation Distinguished Investigator Award. And just only to tell you about the quality of his research, just mention that in his 10 most distinguished publications, he has 3 Nature Structural and Molecular Biology, 1 Cell, 2 Nature Communications, and 1 Molecular Cell. So for that reason, I tell you that he is a very successful researcher. So thank you very much, Guillermo, for coming and visit us. So
thanks for the kind introduction, and thank you for the invitation. It's always a pleasure to come here. Is the microphone okay? Yeah? Okay.
Those people attending by YouTube, you know that you can ask questions later in the chat. And please, if you notice a problem with the sound, please tell us.
Okay, thank you very much. So I'm just gonna give you a talk about our latest results, which involved our studies into the CRISPR-Cas systems. And first, I'm gonna give you a small introduction of why CRISPR-Cas is so fashionable right now, and also why we think that it has been a very important discovery. But to understand the principle, because it's a little bit complicated, I have just made this a slide here, which just shows that CRISPR-Cas is a heritable adaptive immunity in bacteria. And basically what happens is that when you have a first infection, a phage can introduce the genetic material into the bacterial cell. And these systems, they have an integrase called Cas1, Cas2, which is able to cleave this DNA and incorporate these pieces of DNA into a CRISPR array, which later can express these RNAs and Cas proteins that form effector complexes, effector nucleases, which are surveillance complexes, which are in the situation of a second infection will be able to recognize this foreign DNA by Watson and Crick base pairing, and then exert the nuclease action into this DNA, and then stopping the infection. So this has prompted CRISPR-Cas as the tool of choice in genome editing, where the main approach right now is to use these molecular scissors, in which you can recognize a piece of DNA that you want to correct or inactivate, and then produce a double-strand break that the cell will repair using one of these two pathways, which is non-homologous enjoining or homologous-directed repair, and then sealing that break and introducing a piece of DNA in case of the use of homologous recombination, or inactivation of that gene by fusing the two ends of the broken DNA and then eliminating the function of that gene by introducing mutations or nonsense DNA. And basically, this area has been quite active for the last 20 or 30 years, but we were using a different type of protein scaffold in order to make a specific nucleases that will target the sequence of DNA. And those were hominin endonucleases, zinc fingers, and talents. And the problem here is that it was very difficult to redesign these protein scaffolds to recognize specifically a sequence of DNA. For that reason, the arrival of CRISPR-Cas has been a revolution, because by exchanging the CRISPR-RNA of this complex, you can basically change the sequence specificity with high precision. Basically, the major part of the people that I always meet during talks, they know very much about CRISPR-Cas, but they might not realize that all the knowledge or all the information they get is 99% focused on Cas9. But Cas9 is not the most abundant CRISPR-Cas in nature. In fact, there are two classes of CRISPR-Cas. One, and I'm going to start with class 2. This class 2 is a CRISPR-Cas class of systems that contain a single effector protein, a very big multi-domain protein, normally around 1,000 amino acids. And then this single effector protein is the one that makes the effector complexes with the CRISPR-RNA, and then it contains other accessory proteins for the steps of adaptation and maybe expression when you have to process this CRISPR-RNA. But as I'm telling you, this is not the most abundant CRISPR. However, it has been strongly used for different biotechnological uses. One is the production of high fidelity variants that will detect the sequences with higher precision. Also, there has been other members of this class 2 that were discovered recently, and one of them is Cas12a, in which we have been working a lot, and I will mention briefly in the following slides. And the other one is Cas13, which has a problem, which is normally found only in thermostable bacteria, and then it cannot be used for editing in mammalian cells. And Cas9 has been also used to fuse different domains that can make, in the dead Cas9, you just kill the nuclease activity, and you fuse it with other protein domains that can help you to perform epigenetic regulation, transcriptional regulation, and maybe other types of editing, which are base editing and prime editing, that can be quite useful in some monogenic diseases. All this work has deserved the award of the Nobel Prize for Emmanuel Charpentier and Jennifer Doudna a couple of years ago, because of the application of CRISPR-Cas guided nucleases and their finding that they could reprogram this nuclease just by exchanging the CRISPR-RNA. I would say that we are in a CRISPR-Cas era, because there are the number of applications where you can apply this technology is huge, and they can go from human therapeutics to industrial biotechnology, drug discovery, agriculture. There is a huge revolution at the moment. However, Cas9 has some problems that I will discuss a little bit later, and this has prompted for the search of other types of CRISPR-Cas that might circumvent this type of problems. And one of them is Cas12a, in which we have been working here, and I think my last seminar here was about the structure of the air loop. But after we finished that work, we wanted to basically understand the transition state of this reaction. And for that, we turn into a technique, cryo-electron microscopy, by which you can investigate or visualize different intermediates during a reaction. And this video that I have here is just to show you how important it is to basically understand what happens in these intermediate steps of a reaction and all these conformational changes. It is a video from the physics of fluids on the University of Twente, in which you can see that if you do not have data of this intermediate state, you cannot understand really how these two bubbles are being fused. And this is something that happens also in these biochemical reactions. So by cryo-EM, we investigated how all these different conformational changes happen during this reaction by using a mutant that is unable to cleave the DNA. And we have done that in our facility at the University of Copenhagen, which is one of the nodes of the cryo-EM network in Denmark. Basically, in that work, we were able to identify 5 different intermediates of this endonuclease reaction, in which we could visualize how the different domains of this protein were able to exchange in order to cleave the DNA. And also, I will not go further in detail on this, but this was basically, we have been also able to search for other type of nucleases of class 2, which are very small. This one only has around 600 amino acids. And the idea is that you can reduce the size of the protein, because otherwise, when you want to use them for therapeutical applications, the size of the proteins plus the CRISPR-RNA is too big to fit into an adenoviral vector, which is the one that you use as a vector to provide the nuclease to the cells that you want correct. So all this technology has been transferred to a spin-off company from the lab. And at the moment, they are focusing on the therapeutic applications of all these different tools in order to correct or inactivate genes and try to treat some diseases. But today, I'm not gonna talk about all the therapeutic part of the CRISPR-Cas, because I think it is important to realize that even though this therapeutic part is important and is pushing research, CRISPR, especially Cas9, it still has a lot of problems when you want to employ them in disease. And there has been different reports of how toxic this can be when you are treating different type of cells, from induced pluripotent stem cells to autologous stem cells. And the problem does not come only from the way it cleaves, but also from the approach that I just described to you, in which you need a double-strand break to start to initiate the editing. And this is a problem because it has been seen that it can promote chromosomal rearrangements. Therefore, all this is prompting the discovery of new types of tools that might be able to perform this editing without double-strand breaks and without the action of repair, and also basically is to search what nature can offer us, not only for genome editing, but perhaps for other type of discoveries. And class 1 is the other big class of CRISPR complexes that I have not mentioned yet, but it's the most abundant one in nature. Class 1 is different from the class 2 in the fact that it's composed by several, the effector complexes are composed by several subunits. And today I will give you more detail about our work in the type 3 system. The main difference is not only that the effector complex is composed of several subunits, but also that there are some signal transduction ancillary proteins that are involved in the immune response that this type 3 promotes for the immunity, into the bacteria. And it is a huge variety of type 3 CRISPR systems in nature. I'm gonna tell you about the type B, which is composed by these CMR proteins, and it has all these different activities. Cyclic adenylate, cyclic oligodenylate synthesis activity, single-strand DNA cleavage, target RNA cleavage, and in our case, we also found that it has single-strand RNA cleavage, as you will see. And the main characteristic of this system is this subunit, which is called CMR2, which is a common subunit in all these different subtypes, which is a protein of around 1,000 amino acids that contains 2 of these activities. The one that is responsible for cycling these ATPs, and also another one activity in the HD domain that is responsible for cleaving single-strand DNA, and in our case also single-strand RNA. So basically what happens here is that these big complexes are assembled, and when the RNA polymerase is transcribing different messengers RNA, this system will work like a shredder. It will be around, floating in the cytoplasm, and testing by complementarity whether the transcripts that come from the RNA pol match or not the CRISPR RNA. And when there is a match, then the transcript will be degraded by these four sites here in this filament, and then it will be available to test another messenger RNA. And the important detail here is that, perhaps it's better, is that, wait a minute. The important detail here is that there is this part which is critical in the whole system, which is called the tag, which is a part of the CRISPR RNA composed of 8 nucleotides, and this other one, which we call anti-tag. And when there is a match between these two, the cleavage only occurs at the level of the messenger RNA in these 4 sites, and the host transcript is degraded. But if the transcript comes from the transcription of a phage gene, what happens is that this tag and anti-tag do not match. And at the same time that the activity of the single-strand RNA cleavage is activated in the filament, these 2 extra activities will be a trigger on the CMR2 in the HD domain and the PALM domain that will cycle the ATPs to synthesize this second messenger, which is cyclic oligoadenylate. Oops, sorry. This secondary messenger activates another complex, which is called CSX1. It binds to this hexameric complex, and then it will amplify the single-strand RNA cleavage activity. And, oops, sorry. And this basically will induce cell dormancy or cell death in order to avoid the phage infection. But as you might understand, to keep high levels of cyclic oligodenylate is a risk for the cell. So the cell has also developed these ring nucleases, which are enzymes which are involved in the degradation of this cyclic oligodenylate, this secondary messenger, to stop the indiscriminate single-strand RNA cleavage. And this is the work of Rafa Molina, who is now here at the IQF, where he has solved the structure of the CSX1 and 2 of these ring nucleases that I will not discuss with you today. So CMR beta, which is the big complex, contains 6 catalytic centers in 37 polypeptides, plus 1 CRISPR RNA in a 1 megadalton complex. And to assay the different activities, we purified and isolated this complex directly from the bacteria, which was not easy. And this complex, we tested all these different activities with different substrates. And this is important to follow the talk. So this is what I call the CTR, is what is called Cognac Target Rna. And basically this type of RNA will represent the status where the CMR beta is triggering immunity, and all the activities will be switched on. The other one is the one that we call non-target RNA, which is the molecular situation in which the RNA is recognized, but you are not triggering the secondary activities to kill the cell or dormancy. So it's a way to avoid self-immunity. And the third one is an artificial construct, artificial RNA, which we call anti-tag deficient RNA, in which the nucleotides which are responsible of immune or the avoidance of the immune response are not present. And in this activity assays, you can see that basically the cleavage of the target RNA is cleaved in all the situations, in all these 3 situations, while the activity for single-strand RNA only happens in the CTR, and CoA synthesis is the same, and also the new activity that we found, which is the single-strand RNA cleavage at UA denucleotides. So basically, using these 3 different scenarios, Nicolas Sophos solved, in my group, solved 6 different structures using cryo-EM, and basically all these different structures try to recapitulate all these different conformational changes that might happen during self-immunity and the triggering of the immunity. These structures were solved between 3.5 and 2.4 angstrom resolution, and basically the APO structure will contain only the CRISPR RNA. The non-target RNA will be in this type of situation, and this will be the Cognac Target RNA, in which we do not have MPPMP, which is a non-hydrolyzable analog that we included in the following scenarios to observe whether the presence of the nucleotide was inducing additional conformational changes. And also we added a poly-T single-strand DNA to see whether we were able to visualize it on the catalytic site that cleaves the single-strand RNA. And basically, Nicolas was able to solve these 3 structures. These 2 were 2 different conformational states on the same sample, and the same happened in the third case, where the single-strand DNA was added. And basically, this is a scheme where you can see in this cartoon how the structure is depicted here. Our CMR beta had a peculiar subunit, which is CMR7, which you can see here is decorating the main filament where the CRISPR RNA is located. And then you have these 4 catalytic sites that cleave the target RNA in the CMR4 subunits, which are located here, 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 and there. And in the CMR2 subunit, you will find the catalytic site that cycles these ATPs and the HT domain where the single-strand DNA is cleaved. And the most important feature that we could observe in this CMR2 was a loop in this subunit, CMR3, which is the one that associates with the tag of the CRISPR RNA, which we call the stalk loop. And this stalk loop is located in a kind of crossroad in the CMR2 subunit. The CMR2 subunit is composed by these domains, D2, D4, the palm1 and palm2 domain, which is in the back side, and the HT domain. And this stalk loop is making interactions in the apo structure with all these different domains of CMR2. This is a detail with a density map where you can see how some polar interactions are stabilizing the loop in that position. And this is a detailed view of the conformations of the different CMR7 subunits, which are decorating the filament of the CMR beta. And as you can observe, they all seem to have a very similar conformation, where it was a little bit puzzling for us. So how is the bacteria, how is this complex protecting the bacteria in order to avoid autoimmunity, to trigger a reaction that will kill the bacteria or will induce dormancy in the bacteria when there is no need for it? So in the structure with the non-target RNA, we can see, we could observe that when we compare that structure with the apo, in this vector plot, you can see that there is almost no displacement of the C-alphas in the main part of the filament, while in the CMR2 subunit, we could observe a rotation and a displacement, a substantial one, in fact. So, what you can see in this video is how this conformational change affects the stalk loop, as you will see in the next slide. And is that the stalk loop is stabilized in a conformation which is completely retracted and different to the one that you can see in the apo. And this is mainly due to the fact that the CRISPR RNA, the tag, is there, hybridizing with the anti-tag, and then this stabilization of this double strand of the RNA is kicking the stalk loop out of that position, stabilizing it in this, let's see, here around 90 degrees. There is a rotation of around 90 degrees, as you can observe in this plot where we have superimposed both of them. There is also a translation of the zinc fingers, which are involved in the palm domain. So then we could basically attribute that what happens is that there is the target RNA can be cleaved by the active centers in the filament, but the two activities that will induce dormancy and cell death cannot be triggered because somehow there is a rotation, but we do not know yet why these centers are not active. Because in theory, you should be able to trigger activity even though the stalk loop was in a different conformation. So why the stalk loop, how it's controlling that? And this was revealed by the structure on the Cognac Target Rna situation. In this case, we could observe two different conformations when we had MPPMP in the solution. In one, we could observe a situation where the bases that should hybridize with the tag were not visible at all. And in other of the conformation, we could observe them, but they were not making Watson and Crick base pairs. They were contacted another side of the CMR2 subunit. And what we could observe is that the presence of the difference on the tag and anti-tag, the non-hybridization in that region, plus the presence of the MPP-EMP induce a further displacement and a further rotation of these domains, suggesting that in the NTR state, the complex is primed, these activities are primed, but they're still not ready for triggering the catalytic activity. And this was revealed by the fact that you can see it here. This is, I have shown you this before, where you can see that the activity is triggered when these bases are non-hybridized. But in the case that when we do not have this anti-tag there is no activity in any of the secondary enzymatic activities of CMR2. So why? So that means that you need to have the anti-tag present, because otherwise there is no activity. But how is this working? And this came somehow was revealed by these structures of the MPP-MP in the state 2 and also in the poly-T structures, in which we could observe that part of the hybrid that runs up the filament, and let me see here, could not even be visualized in some of them, showing that there is some displacement of the D2 and D4 domains interrupting this hybrid. And this probably can be seen better here in these two movies, in which you can observe this will be the conformational change between the APO and the MPP-MP1 conformation, in which you can see that because the hybridization happens and these extra 4 bases can be visualized, the loop is not visible here because the bases are there, while in the second conformation, which goes between the MPP-MP1 and the MPP-MP2, you can see that this movement is also emphasized by the D2 and D4 domains, which are pushing towards the double strand of the RNA, distorting this part of the hybridization with the target RNA. And these two sites match with the last 2 active centers that cleave the single-strand RNA, CMR3 and CMR4. And I would like you to keep this in mind because I think it's important for the biochemical experiment that comes later. So we could observe that there was this change of conformations, and that the stock loop was basically like the stock of a cuckoo moving from one conformation to the other. We could also observe that the cyclic oligoadenylate activity, we think it is regulated by the stock loop, because in the NTR situation, well, first of all, we could see that for the entrance to the catalytic side that cyclics the ATP, there are 2 entrances, entrance A and entrance B. And in the NTR situation, the one that avoids self-immunity, one of the entrances was occluded by the movement of this stock loop. And the other one, you can see it here. So we deduce that this new conformation or this stabilization of the stock loop in the NTR will avoid the entrance or the exit of this secondary messenger and probably will hamper catalysis. And in the case of the HD domain, which is activated by the conformational change, we could observe that these changes between the D2 and D4 basically position the amino acids in the catalytic side into a conformation where they could start to do single-strand DNA cleavage. So in order to understand how the target single-strand RNA can be coordinated with the triggering of these two activities, we perform an experiment with different oligos that contain the anti-tag and different lengths, because we wanted to check whether this conformational change of the D2 and D4 domains were related to the triggering of the activity below here in the CoA and the HD domain catalytic sites. And what we could observe is that all these short target RNAs were able to, the RNA was cleaved in all the situations, but only when the substrate had enough length to reach the second catalytic site here, CMR42, and have some bases extra, the activity was triggered for the single-strand DNA and also for the CoA synthesis, indicating that in order to accomplish all the conformational changes that will lead to activity, you need to have at least a cleavage on the target RNA up to this level. So there is a coordination from the lower part of the filament with the conformational changes that you see in CMR2 in order to trigger these activities. It's collectively is a kind of a concerted action, allosteric activation due to the cleavage of the target RNA and the non-hybridization of the 5' tag and the 3' anti-tag, which controls the activation of the CoA and the HD. And also the presence of this extra nucleotide here is necessary in order to activate the CoA activity, because they interact with the zinc fingers of the PAM2 domain. So after this, we wanted to understand what was the function of CMR7. And basically, CMR7 seems to be a regulator of the activity of the complex. But we also think, and we couldn't do this experiment, we think that this is a kind of anti-CRISPR protection. And there are some phages and bacteria are in a continuous fight during this evolution to see if phages want to infect the bacteria and bacteria wants to protect themselves of this infection. And there are some proteins which are called anti-CRISPR, which are produced by the phages, that basically what they do is that they bind to the effector complexes of CRISPR to avoid that they can cleave the DNA of the phage. And our hypothesis is that this complex, this CMR7 subunit of the complex, may be a protection to avoid that anti-CRISPRs bind to the effector complex. So basically, in this work, we have find out what is the difference between the self versus the non-self recognition mechanism. Basically, to avoid autoimmunity, what the complex does is to fix the stalk loop in a position where ATP cannot be used to synthesize CoA, and there is no single-strand nucleic acid activity, and then the complex is locked in that situation. But it is very similar to the case where you're gonna trigger all these secondary activities. The difference is that in that case, where you will trigger the immunity and you will synthesize the secondary messenger, what happens is that the loop is flexible and is alternating between all these different situations. And then the conformational change that we have observed in the D2, D4 domains can happen. The activity on these two catalytic centers can also proceed, and then all the secondary activities are triggered, and the secondary messenger is released. And then what happens? Well, what happens is that, as I mentioned in this slide, this binds this CSX1 complex that is going to amplify the RNA cleavage. And basically, this is the work of Rafa. This is another big complex. Well, not as big as the other one. This is around 300 kD. And the curiosity of this is that we solve the structure combining cryo-EM and X-ray. And this is because it was impossible to solve the phase problem for this protein complex, because we purified directly from the bacteria, so we couldn't use selenomet. And to do the heavy atom derivatization was not working. And molecular replacement, there were no proper models to do. So we got a low resolution map at home, and then we use this map to phase the crystals by molecular replacement, solve an initial structure, and then get high resolution structures of different conformational intermediates. And basically, the structure is an hexamer, which is composed of 3 different dimers. The protein contains this carf domain, HTH, and EPEN domain. This is the one that binds the cyclic oligoadenylate, and this is the catalytic RNase domain. And basically, they are located on different sides, on the proximal and the distal side of the complex. And this is, for example, in the hexamer where the cyclic oligoadenylate binds, and this is where the RNA is getting cleaved. And at that time, we didn't have, there was no commercial cyclic oligoadenylate, so we produced that ourselves by using another type 3 complex, which is called CMR beta. We basically purify this complex and use it to synthesize the cyclic oligoadenylate and purify it. And this is the work of Minxia and Irina. And then we were able to get crystals with the compound, and we could visualize how this cyclic oligoadenylate was bound to this indiscriminate RNAs. And because this is a thermophile, we were able to see 2 different conformations when we crystallize it at different temperatures. And we could validate this by difference and omit maps, that there was a change on the conformation of the ligand when bound to the in the catalytic side. No, sorry, not in the catalytic side, but in the ligand binding side. And basically, we could observe these 2 different conformations, and most interestingly, between the conformation 1 and the conformation 2 of the ligand, we could observe that those conformational changes were accompanied by a conformational change that was transmitted through the protein in order to position the catalytic site here for the cleavage of the RNA. We also investigated whether there was a cooperativity in this binding, and we could observe that there was a very clear positive cooperativity on binding that was accompanied by an increase in the activity. So we could understand now how the second messenger is produced, how this complex avoids the production of this second messenger when a cellular transcript is captured, let's say, and we could now understand how this cleaves the single-strand RNA to induce dormancy or cellular death. And we have also the structures of the proteins that cleave this second messenger to reduce the levels, but this I think Rafa can tell you because he is the one that has solved that. So I think I'm not gonna discuss that with you today. So so before I end, I would like to not only acknowledge all the people involved in this work that I have acknowledged during the talk, but also I would like to mention that the study of these systems, which in principle is pure basic science and we don't know where they're gonna lead us, they can bring up interesting opportunities in different areas. At the moment, we do not know how this can be applied, but I think it is worth to continue in this research line, because all these studies have led to revolutions, from restriction enzymes to the discovery of CRISPR. And now, in this case, I was mentioning to you before the talk, this system has shown that the pattern of innate immunity that is well known in human cells, in mammalian cells, is somehow conserved in prokaryotes. So prokaryotes, they already had developed an ancient innate immunity system like this one. In mammalian cells, it works in a similar way, synthesizing a secondary molecule that is triggering an RNase. So I think we can learn quite a lot of all these systems, also for the evolution of our own immune system. So thank you very much for your attention, and I'm happy to take questions.
So thank you very much, Guillermo, for this awesome talk. And I think it's time for questions. So please go ahead. So let me tell you just one comment to start. After attending your talk, it is not clear to me if you are a structural biologist, a biochemist, or even a microbiologist. So from a methodological point of view, your group did all work that you show here,
Yes.
or do you have a network of collaborators?
No, all this was done in our lab.
So you are a structural biologist, biochemist, microbiologist?
Yeah, I think we tried to cover my part of that. Our only collaboration on this was the people that providing us with the complex, because this had to be cultured they had to be cultured in a huge containers with high temperature. But all the rest has been done in our lab.
So, okay. Amazing! Congratulations. So
we try to. I mean, I think in the area where we work on is not only this. I think there is no reason to I would say divide science by technologies. I think you need to to understand what you need to try to push for your aims, which is to understand the system, and then you apply what you think is needed.
Yeah. This is possible because you have all the facilities to carry out these different.
We have a good lab, and then the setup of the cryo-EM, I think, has opened a wide area that goes beyond looking at the structures.
So please, Pablo.
Guillermo, nice talk. I mean, you show us a very nice example of the dynamics of this system. But I suppose this is the complexity or the dynamical complexity of this system could be wonderful. So, could you comment more about other motions, or what are the main motions that you observe just by cryo-EM?
Yeah. I think the main motions that we can observe, this is a kind of we can observe many different motions, but as you can see on our case, I think it was, let me see. As you can see, basically when you see these vector plots, this is referred to the coordinates of the C alphas. So basically, we could observe all these movements, and sometimes, depending on the resolution, you can see more fine detail. for me, the most important thing on all these motions is how you can use them to interpret function. And in our case, you can say that we have used a trick, which is trying to mimic, in this case, different biochemical conditions by providing different substrates. Okay, and I think this is a valid approach. In some cases, you need to test it with activity assays. In other case, like in one of the papers that I had in my introduction, in this case we use only a single mutant to capture all the different conformational varieties that we saw. In this case, we needed different approaches because the sample is much more complex.
Suppose you also focus in a given state and compare this.
Yeah, we started from the apple and then go to the rest. Yeah.
My question was, I mean, the heterogeneity of your sample in probably
This was not the most heterogeneous sample that we have.
Okay.
We have other cases where heterogeneity is huge and you cannot see domains. And the problem there is to justify, because in some cases you have in the same sample, you can see many different conformations. And in some cases you cannot even visualize some of the domains. And this you have to be very careful because it depends very much on how you make the particle selection. And yes. And then, but I think it is very important, the information that you get there for function. So you can dig more and more and more. In this case, this was not a very heterogeneous sample. We have much worse cases. So this, in fact, is a good case. It was an easy case to because the conformations of the loop were clearly seen, and you could. Yeah. I think the main problem is that in some cases, for example, when you see that there is no density for something, then you need to support that with other experiments to make sense of that conformational change.
Thank you, Guillermo. It was a wonderful talk. Congratulations. I have a probably naive question, but in the hybrid you saw of that recognize the protein complex, this hybrid is not duplex; it's nearly flat, no? So
it's not it's not
helical.
Yeah.
So basically, that force as a base every 3, 4, or 6 bases sticking out of the pairing, no?
If there is one, I haven't gone into detail into that because the talk is too long. But there is one base that is flip out by one of the domains.
Exactly.
One of the in this CMR4 subunits is flip out.
And I noticed that base is a purine. It has to be a purine all the time, or not?
No, I don't think so. I don't think you need to be a purine. No. No.
Okay.
No. I think the main specificity or the main region where the specificity is key is in this tag anti-tag region, and it is quite. This sequence is 8 nucleotides, but 4 of them are trapped by the interaction with the. Let me see. 4 of them are trapped on the. No,
see, on that one you can.
On CMR3. So out of these 4 are trapped by the protein in order to make the complex, because what happens in the cell is that you can have CRISPR RNAs, this I haven't mentioned, you have CRISPR RNAs of different length. And this filament here can change. Because then you will assemble the CRISPR RNA with CMR3, and then it will grow depending on the length of the CRISPR RNA. And because these changes, we have also structures of complexes with 2 or 3 only of these catalytic sites. But the key point or the surprising point is that basically the whole thing depends on 4 nucleotides. 4 nucleotides that are here for the hybridization, and I think this is why the activity of these 2 catalytic sites and the interaction with the D2 and D4 domains is key to trigger the whole activity for the secondary messenger and the cleavage, because otherwise it will rely only in the hybridization with 4 nucleotides.
But the question is, another question that came to me is, the cleavage site is near this stick out base or not?
No, it's over there. I mean, I don't have it here, but it's in the near, is in the neighborhood, but I don't think it's involved in the cleavage. Yeah.
Sorry, maybe wait. The signal is quite nice because it's something that you detect from the bottom to the top and then goes up.
Yeah.
Okay, thanks.
Thank you. Well, in a way, you have already answered my question. Is why is so big and why is so complex? And you said that could be different lengths.
Yeah.
But does it require a very tight regulation, or?
There is a very tight regulation here, which we don't know much, because, for example, in this bacteria, you have the CMR beta, but there is also CMR alpha that we have solved the structure to. Basically, CMR alpha is similar to this guy, with the difference that there is no CMR7, and the biochemical property that it synthesizes much more cyclic oligodenylate than this guy. So how all these things are regulated is not known. It's the same for the enzymes that cleave the cyclic oligodenylate. There are 2, and we know that one is much faster cleaving than the other one. So we have the structures. We know basically we have all the structural picture of the pathway, but we do not know yet how the cell regulates all these systems and the levels.
Would it be post-transcriptional regulation?
We don't know. Yeah, we don't know. Oops.
More questions? Is there any question on YouTube? Is there? No. I have another comment or question about the evolution of these systems. As far as I know, I saw some numbers about bacteria and archaea in which these systems are present, and it seems that approximately in the half of the genomes that has been characterized of bacteria, coming from bacteria, these systems are present, and in more than 80% of archaea have these systems.
Yep.
So do you expect there are other immune-like systems in bacteria?
Yeah.
Other than this type?
Yes, there are many.
Oh, really?
Yes. There are many different. This is one. CRISPR is one of the immune systems, but there is a huge variety of. When I moved to Spain, I thought bacteria was dead. There was nothing else to do in bacteria. Now, when I went back to revisit bacteria, there is plenty of immune systems. There are retrons, there are UDP, there are plenty of immune systems that are trying to fight these mobile genetic elements, phages or plasmids that can infect. And you have to realize that phages are the major living entity on Earth. So there is a huge, I would say, evolution pressure. Yeah, and there's a huge melting pot there, trying to fight that. So there is plenty. Yeah. I think the good and the fact that this is predominant in archaea, I think is a good example of how, especially for the correlation with innate immunity in mammals, that all these pathways were already present there. So we hope that this might give us some hint on how the immune innate system in mammals work.
This is surprising. And since you are quite very interested in kinetics of processes and intermediates, and I would like you to explain, because it is not clear to me. I see that when, let's say, vaccinated bacteria or archaea against one virus contact these viruses, it fight against them. But what happens at the beginning when it is not vaccinated?
Yeah, that's a
Which is
good
the
question.
kinetics of these processes? Because
It's not clear how immunity, this initial step of immunity is this initial step, we know how it works at molecular level. We know we have this Cas1, Cas2 complex is able to recognize this material, but we don't know how this has been originated. And we don't know what happens when a new phage comes there. What is the kinetics? Yeah. So what we know about this process is that there is one sequence which is called PAM, protospacer adjacent motif, which in fact is I think it was named by Francis Mojica. And in this PAM system, this PAM, which is a sequence of 3 or 4 nucleotides, usually is left out of this sequence that is incorporated in the CRISPR array. And this is done in a way because this complex does not include it when it cleaves and integrates. And this PAM is essential because is the one that it works as a label for these surveillance complexes. Because first they recognize the PAM and they test hybridization. And I mean, if there will be PAM if PAM will have been included here, then it will cleave its own genome. Okay? But how this is acquired here is a question for the first time. For the second time, once that you are naive, for the first one, when you are naive, this is still something that is not clearly. I guess it can be that some cells die, and one possible explanation that just came to my mind, maybe that in the first infection you kill the cell, and then when the cell is lysed, the other cells can acquire some of this material.
Exactly.
And then they have the immunity. But the precise way that happens is not known.
Okay, thank you very much. So I think there are no more questions. So thank you very much again. And as always, we have a small present for you for participating in our seminar. So thank you very much, Guillermo.
Thank you for the invitation. Thank you. Good.